So we've got our DNA molecule now. All the nucleotides are connected to each other by the phosphate bridges, and the two strands are lined up anti-parallel, one going one way, the other going the other way, and the hydrogen bonding between the bases are kind of holding those strands together, and the weird angles of the rungs of that ladder causes the molecule to twist into that famous double helix shape. The problem, though, is that we still have this 3 billion base pairs molecule, 6 feet long about if you stretched out one molecule of DNA. And we've got to pack it into a very tiny space in the center of your cells, the nucleus of the cell. 6-foot molecule, 3 billion base pairs into a tiny, tiny, tiny little space. Well, we've got to fold it up. Folding a molecule this long has to be done very carefully, because if you just crumple it up and just kind of squish it into a ball, that's like taking a piece of thread, 2 pieces really, 2 strands, a mile long, and crumpling them up into a ball, and then expecting that you're going to be able to pull them out again without them being tangled. We can't let our DNA get tangled, because if it gets tangled, we can't use it for anything. We can't get any information out of it. So we have to fold the DNA, and it has to be done in a very, very ordered way. Now, there aren't little guys in your cells working on the DNA and folding it up. The DNA folds because of its structure. This is all chemistry. It just happens because of the way the molecule is structured. But there are some helper molecules that are going to come along to help fold it. To start with, we have to fit it into the nucleus. The nucleus is this tiny little area. And so what we're going to do is we're going to try to organize it a little bit. If you have a nice long piece of thread and you want to organize it a little bit, you spool it, right? You coil it around a spool. Well, we have the same kind of thing happening in our cells. We have these tiny little proteins called histones. And the histones act kind of like a mini spool. The DNA wraps around each histone twice. And it's held there by attractions between the DNA molecule and the surface of the histone. They're like attracted to each other, like magnetic attractions. It ends up making your DNA look like beads, the histone proteins, on a string. And each one of those beads, the histone with the DNA wrapped around it, is called a nucleosome. There's been some really interesting work in looking at these nucleosomes, and specifically looking at the distance between them on the strand of DNA. It turns out everybody's got their own unique distance between their nucleosomes. And that makes for some interesting ways of identifying people instead of looking at gene structure, but that's something else. When you mix these proteins with the DNA and you throw in a little bit of RNA, you get something called chromatin. Now, chromatin comes from the Greek word for color, chroma. And chromatin literally means colored material. It was called this because the first time it was seen under a microscope, it had to be stained, and so it had a bit of color to it. When you're going to separate the DNA and move it around a little bit, normally DNA just kind of is wrapped around these histones and just sits kind of as a loose clump in the nucleus, and it can't really be seen. It's just sort of this sort of colored mess. But when it's time for a cell to divide, you have to copy the DNA, and then you have to separate the two halves of DNA, the two copies, into separate cells. And in order to move a molecule that's so big and so tangly, we have to condense it even more. And so when DNA is condensed into very compact structures, they're called chromosomes. So chromosomes are made of DNA wrapped around the histones, and then there's some other proteins that sort of give them the shape, that sort of sausage-like shape that you're familiar with. And there's some RNA in there that helps to regulate things and make sure that it all stays together. And that's what chromosomes are. But you can only see chromosomes when we're going to be moving large pieces of the DNA from one side of a cell to another, when the cell's getting ready to divide. So here's a picture, sort of how DNA might be coiled. And down here at the bottom, we have the DNA coiled around these little balls. That's the histone. And then those themselves are coiled up to make a coil. And then that coil is coiled on itself. This whole structure is called a supersolenoid, a coiled coil. And finally, that's sort of set onto this protein scaffold to make a chromosome. that can then be moved. So we've got this long molecule that we just continually coil and coil, and we coil the coils until we have a nice, dense, packed, but ordered way of moving the DNA around. This is an electron micrograph, a computer-generated electron micrograph of a histone protein with the DNA wrapped around it. The black and white structure here is the DNA. You can see it's sort of wrapped around the colored structure, which is a histone protein. The histone protein is actually made of 8 separate mini proteins that come together and act as a unit. And we'll talk more about proteins later. But that's kind of what a histone looks, and your DNA molecule is wrapped like that around countless of these histones. And then all of that is coiled up and coiled even further to make your chromosomes.